Hello and welcome to Lesson 3, Enzymes with Mrs Castanera. This is the third lesson in the GCSE organisation topic, and we're going to start today by looking at the key words. So our key words today are enzymes, amylase, protease, lipase, soluble, insoluble, protein, lock and key, and specific. So we're going to highlight those, please. By the end of the lesson, you will understand what all these words mean, and they are explained and discussed throughout the lesson. So next we have the content of the specification, which tells you everything you should be able to do at the end of this topic, ideally at the end of this lesson. And so what I'd like to do first is start by completing the knowledge check on page 25, answer the questions up to 8, and then we'll go through those together. Pause the video whilst you do that. Okay, let's go through those. So, question 1, state the function of carbohydrates. Well, carbohydrates provide energy. So use another coloured pen to tick it to show you got it right, or write in your correct answer. Question 2, state the basic function of proteins. Okay, so the phrase you need to remember here is growth and repair. When they ask you about what proteins are for, they are for growth and repair. Question 3, state the function of lipids. Remember, the lipids are fats. They are for energy storage and insulation. If you've got either of these, you can have the mark, but you'll need to write the other one in as well to make sure you understand that both of them do count. Question 4, state what glandular tissue produces. So glandular tissue is the tissue of a gland, and that produces hormones or enzymes. Again, I will accept either answer for the mark, but please do make sure you write in anything you've missed. You will need to write both down, please. Question 5, define peristalsis. So peristalsis is the movement of muscles, so bands of muscles contracting to push food down the esophagus. So it's the idea, it's about the contraction of muscles or squeezing of the muscles to push the food down the esophagus and into the stomach. If you're not sure that your answer is quite good enough, make sure you write out the full answer next to yours or on the notes page if necessary. Question 6, true or false, food enters the pancreas. So we should know from previously that no food enters the pancreas. Okay, so that is false. Things are only emitted from the pancreas, things are only given out from the pancreas, nothing enters it. Question 7, state the function of the liver. So the liver filters blood, it detoxifies chemicals, and it metabolises drugs. Any one of these is sufficient for the answer, but again, do make sure you write all 3 of them down, please. Question 8, state what the essential nutrients are. Okay, so our essential nutrients are carbohydrates, proteins, fats, fibre, vitamins, minerals, and water. In fact, sometimes they talk about essential nutrients, they mean things we can't produce ourselves or essential to keeping us alive. Now, fibre and water are very essential to keeping us alive. Sometimes you'll see fibre and water not included on this list, but they are essential to life, and they will generally talk about the 7 essential parts of your diet, and this is that. This is what you need to answer for those. So please make sure you've got all of these down, okay, and add anything you've missed. So if you give yourself a mark out of 8 for this, please. Okay, make sure you've recorded everything down, and then we'll move on to the next section. Okay, so today's lesson, we're going to be describing how enzymes work, so you'll be able to do that by the end of the lesson. And we're going to state examples of different types of enzyme, what substrate they work on, and where they are found. And we'll be able to do all of this by the end of the lesson and understand each of these key terms that's being used. So, digestion and enzymes. As discussed last lesson, animals need nutrients from food to function. In order to get these nutrients, food needs to be broken down into smaller components. So that's what digestion is, it's breaking down into smaller components so we can absorb it. However, if the body just left the nutrients to degrade by themselves, then it would take a very long time. Enzymes are special chemicals in your body that speed up chemical reactions. So we know this from last lesson. I'm just going to add a little note Here, so this is digestion. One of the reactions these enzymes speed up is the breakdown of large food molecules. Okay, so enzymes speed up the breakdown of large food molecules such as starch, proteins, and lipids. Enzymes are a bit like biological scissors, and we'll talk about them in this way. So they cut things up. They break the bonds between the molecules. Now, enzymes are found in different parts of the digestive system, and this table below shows us where each of these enzymes is made, is produced, and where they work. Okay, so here we have the large molecules such as lipids, proteins, and starch. So lipids means fats, and starch is basically our complex carbohydrates. So things sounding like pasta and bread and that. The small molecules, okay, is what they turn into. So draw a little arrow to show that they turn into those after digestion. And so lipids turn into two molecules, fatty acids and glycerol. Proteins turn into amino acids, and starch turns into glucose. Now the enzymes used to do this. So for lipids, the fats, it's the lipase enzyme. For proteins, it's the protease enzyme. You'll notice lipids, lipase, they both begin with a LIP. Proteins, protease, they both begin with PROT. And starch, which is a carbohydrate, is the carbohydrase enzymes. Now there are examples of each of these enzymes. So a type of protease enzyme is pepsin. A type of carbohydrase enzyme is amylase. And this is where each is produced. So the fats, the lipids are produced, the lipase enzyme is produced in the pancreas. So we know that fats are digested after the pancreas, so in the small intestine. The proteins are digested after the stomach, or in the stomach and pancreas, or after the pancreas even. And that's where the protease enzyme is produced. And starch is digested and turned into glucose from the very first moment it enters the mouth, because the carbohydrase enzyme is produced in the salivary gland as well as the pancreas. Okay, so you'll need to learn this. You need to know all of this information. So, for example, if I were to say, where is protease produced in the body? You need to know the answer. If I were to say, what is starch broken down into? You need to know the answer. You need to know that it's glucose. If I say, what breaks down lipids into fatty acid and glycerol, you need to know that that's the lipase enzyme. Okay, so make sure you spend some time learning that. Feel free to pause the video so that you can do this. Okay, so we're going to move on to how enzymes work. So enzymes have a three-dimensional shape, a 3D shape. Okay, that's really, really important. Okay, so they're very much a 3D thing. And the shape includes like a groove, which we call the active site. Okay, and on this picture below, you can see this groove shown here. Now, they don't really look exactly like this, but it's very similar. And that's the active site. And the molecule to be digested is called the substrate, which you can see here. This is the substrate. And that is this sort of yellowy orange molecule here. And our enzyme with its active site, you can see here. So we've got our enzyme in green, our substrate in yellow, and the active site, which I've highlighted here on our little green enzyme. So the way it works is the substrate fits into the active site, okay? A bit like a key fits into a lock, okay? So we compare it to a key fitting into a lock. That's why it's called the lock and key explanation, okay? Or the lock and key theory. So we call this the lock and key theory, or idea. Just zoom out so you can actually read all that. Okay, so we call this the lock and key theory. So a key will only fit into a lock if it's the correct shape, and that's really important. So the substrate will only fit into the enzyme. So the substrate's the key, the lock is the enzyme. But let's annotate that. So the substrate is the key and the enzyme is the lock in this analogy. Okay, and so this substrate key is only going to fit into the enzyme lock if it's the right shape for the enzyme. The lock can't change the shape it requires, so the enzyme is built with its particular shape that it requires. However, it can take only one type of substrate, only the substrate that fits into it. Okay, so we're going to highlight this here. The enzyme is the lock and the substrate is the key. Now, enzymes are referred to as biological catalysts. And remember, a catalyst is a substance that speeds up a chemical reaction without being used up itself. So we need to remember what a catalyst is, that's very important. This is a definition you are often asked for in exams. Now, enzymes speed up all sorts of chemical reactions in your body, such as respiration, okay? So turning oxygen into energy and producing carbon dioxide. And digestion, where we take large food particles and break them down into the teeny tiny small molecules that can actually be absorbed. Enzymes do not get used up, okay? That's not possible. Okay, so enzymes work again and again and again. In the same way that the lock on your front door works again and again and again. You can put keys into it hundreds, thousands, probably millions of times, okay? Same thing works for enzymes. Now, we talk about enzymes being specific, and that's because, as I said earlier, they only work, this lock only fits one specific key, okay? So the enzyme will only fit one specific type of substrate, and that's why we call them specific, because only one molecule type will fit in there. And so this is what happens. So our substrate comes along, and this could be some protein, it could be some lipids, it could be some starch, and it's going to come along and it's going to fit into our lock, the enzyme. And this is called the enzyme substrate complex. Here we have that. And the next thing that happens is we get the enzyme back, so back to the start with our enzyme, but now we have products, more than one. So we can see that there is a gap down the middle here, and this enzyme substrate complex, the enzyme is broken down the substrate into two pieces, or sometimes more, okay, and it has broken the bond attached these two pieces together, and so we've got two separate pieces left. A bit like if this was our protein and now we have amino acids, or if this was our starch molecule, now we have some glucose. And that is exactly how enzymes work. So because they're specific, these enzymes, it means that each enzyme's active site has a specific shape for the substrate involved. Okay, so each kind of molecule that needs to be broken down into smaller pieces by the enzymes, they have their own specific enzyme to do that. Okay, because each enzyme only comes in one shape. And that means we need many, many different enzymes, a lot of different enzymes, because there's so many different substrates in the body. So many different enzymes are needed for the many substrates that are involved in the reactions of the body. So we, of course, then have a lot of enzymes in our body. We're all going to write this down, please. So we have a lot of enzymes in our bodies. What if they don't work? Well, that's a good question. So enzymes, they work within a very specific optimum condition, okay? So it's very important to know that enzymes will not always work. They will only work at their optimum, and different enzymes have different optimums. And if the conditions, such as temperature or pH, change, then we're going to have some problems. So they work within specific optimum conditions, and they are affected by the conditions that vary from these. So we're going to put some examples of conditions. So temperature, pH is another one. Okay, so for example, enzymes work best at body temperature. So body temperature is 37 degrees Celsius, and that's where enzymes work best. If the temperature is lower, they work more slowly. For higher temperatures, we have another problem. So low temperatures mean they work slowly. High temperatures will change the shape of the active site. Now that's a big problem, okay? This process of changing the shape is called denaturing. The enzyme becomes denatured. Okay, so it's not good if the enzyme becomes denatured because it no longer fits. So it won't work, it will no longer work because the active site is the wrong shape. So it stops working, it's the wrong shape, the active site, and it no longer fits the substrate. Okay, so the enzyme is said to be denatured, just as we wrote a moment ago. So this process of denaturing can occur when enzymes are at the incorrect pH. So if you change your conditions from acidic to alkali or vice versa. Now different enzymes have their own optimum pH. Now most enzymes will work at the pH of water because much of our body is made of water and most of our body is pH 7. However, the digestive enzymes don't always do that. So for example, protease is an enzyme that's found in the stomach, okay, and it has a pH of 2. Okay, because of the hydrochloric acid in the stomach, so it has to work at pH 2. So it will denature as soon as it enters the small intestine, where the pH is ever so slightly alkaline at 8. Okay, so this is a problem for protease. So protease only works in the stomach. Okay. Now, what I want you to do next is we're going to watch these videos. Okay, so you're going to watch both of these videos, please, both of them, on YouTube. Follow the details carefully, or if you've got a digital copy of this, which you should, then click on those links. Then what I want you to do is have a go at answering this question here about trypsin, which is specific to catalyzing one particular type of reaction, okay? So you're going to talk about trypsin, okay, and you're going to use the lock and key theory, and you're going to explain what is meant by enzyme specificity. So there's 3 marks there, okay? So you have to write 3 very different separate things. So watch both those clips and have a go at the answer. You may need to re-watch parts of the clips to help you do that. That's fine. This will take you a little while, okay? So pause the video whilst you do that, please. Okay, so by now you've watched both those videos and you will also have answered the question below. If you haven't, please pause the video again whilst you complete the checkpoint question about trypsin. If you have completed it, then let's mark it together. Okay, so your answer can include any 3 of the following statements. So the idea that the enzymes and the substrates fit together, so like a lock and a key. So either of these statements works. The idea that enzymes have a specific or a unique active site, so you could talk about the shape of the active site is particular to the enzyme, anything like that. Then the third part mark would be for saying that it fits with the shape of the enzyme, it's complementary to or fits with the shape of the enzyme, or the enzyme only fits for a particular substrate. Then another mark available is the idea that they bind together to form an enzyme substrate complex and that the molecule gets broken down. So for example, if you had drawn the diagram we previously had on page 26, this diagram, you would essentially have got full marks for that question because it's out of 3, as long as you'd sort of put the point or annotated the points about that these are like the lock and key. Okay, so give yourself a mark out of 3. It was a tricky question, so don't worry if you didn't get all of them. What I do want you all to do is make sure you've written all 4 of those points out, okay? So if there's anyone that you haven't got, please add to them. There's plenty of space below to write those in, okay? So I'll leave that for you to copy that out if necessary. Pause the video whilst you do that, and then we'll move on to the next learning activity. Okay, so we're going to have a go at the exam questions, learning activity 2. So here, all you need to do really is just have a go at the exam questions. Okay, it's everything we've covered during the lesson and previously. Now, there's a couple of pages of these. Okay, so there are 2 pages of those, so continue on to the next page. Pause the video whilst you do this. Okay, again, it might take you a little while. You may need to have to look back through the booklet to help you answer things, or even rewatch some of the videos if you feel that will help. Feel free to look back to previous lessons if you think that will help as well. Have a go, see if you can do it yourself, or look up the answers yourself if you get stuck. We'll mark them together once you've had a go at that, so pause the video whilst you do that. Okay, let's go through those then. So question 1, the body uses enzymes to digest or break down large food molecules into smaller molecules. Draw one line from each large food molecule to the enzyme that acts on it. So if you recall, this was from what I told you to memorise earlier. So starch is digested by the enzyme amylase. So tick it if you got that correct. Fats or lipids are digested by the enzyme lipase. Again, tick it if you got it correct. Proteins are digested by the enzyme protease. Okay, so there should not be anything attached to this isomerase. Okay, so I don't want to see anything attached to that. So if you've done as above, you get 3 marks, so circle the 3. Otherwise, please write in how many marks you got in the corner here. Next question. So draw a ring around the correct answer to complete each sentence. So starch is broken down into sugars. So tick it if you got it right, correct it if you didn't. Fat is broken down into fatty acids and glycerol. Again, tick it if you got it right or correct it if you didn't, and make sure you're using a pen of a different colour. Third question, protein is broken down into amino acids. So again, tick it if you got it right, correct it if you didn't. And next to this, I want you to show me how many marks you got out of the 3. So put that in a circle to show how many marks you got. And then we're going to move on to the next page, which you should have answered already. If not, please pause, answer the questions that are remaining, and then come back to joining us marking the answers. Bile helps digestion. Where is bile produced? Draw a ring around one answer. Well, we should know that bile is produced in the liver, so tick it if you got that right, correct it if you didn't, circle the mark, and then it gives us a total of 7 marks. So underneath that, I want you to give yourself a mark out of 7 for question 1. We're going to move on to question 2 now. Please make sure you've answered this or attempted to answer this. Even if you're not sure, make sure you've written at least one answer on each line, because it's always worth having a guess even if you're not sure. Okay, so let's go through question 2 now. The diagram shows the digestive system. Complete the following sentences about digestive enzymes. Amylase works in the, so you can say mouth or small intestine, either of those is acceptable. Where it is involved in the digestion of, you can write starch or carbohydrate, either of those is acceptable. 2, sugar or glucose, Again, either of those is acceptable. Okay, so there's a mark out of 3 for that one. Next part to the question. Lipase works in the pancreas or small intestine, either is acceptable. Where it is involved in the digestion of lipids or fats, either is acceptable again. 2, fatty acids and or glycerol. So if you've written either of those, that's good. I would prefer it if you'd written both, and if you haven't written both, please do write the second one. You can still have the mark for writing one, but I want you to make sure you've got both written down, because you need to know that it's broken down into both, because sometimes they will require both to be written for the answer for the mark. So again, give yourself the mark out of 3. And we're going to move on. Which gland produces amylase? So that would be the salivary gland, or the pancreas, or the small intestine. Okay, all 3 of those produce it, so any one of those 3 is an acceptable answer. And lipase is produced in the pancreas or small intestine. Again, either one of those is an acceptable answer because it's produced in both. So give yourself a mark there. And then at the bottom, it shows it's out of 8, so give yourself a mark out of 8, please. Okay, so once you've finished that, and I'll just put that back so you can all read the answers and do any corrections you need to. Pause the video if you need that, because that's going to go in a second. Okay, so we're going to move on. So next I'd like you to attempt the knowledge check on page 30. There are 10 questions, so I want you to pause the video whilst you have a go at the knowledge check, please, and then we'll come back to it and mark it together. Okay, so let's go through those. Question 1. State the definition of a catalyst. So a catalyst is something that speeds up a reaction without being used up itself. So there's 2 parts to this. The first part is the speeding up of the reaction. Okay, and the second part is that it's not used up itself. Okay, so it's 1 mark. You do need to write both things for the 1 mark. Okay, so please make sure you have both. If you don't, please add them, correct it. Give yourself a tick if you've got both, and make sure you've got all of that written down, please. You should be using a different coloured pen to mark. Question 2. Suggest why enzymes are called biological catalysts. Well, they're catalysts and our bodies make them. So they're biological because they're made by our bodies. They're made by biology. So we make them ourselves. So anything saying that they're made by living things. Okay. Question 3. Suggest why enzymes are called specific. Well, that's because they only work on one type of molecule. Okay. So if you say they only work on one specific molecule, one specific substrate, one specific shape, or one enzyme only works for one thing, that's fine. Question 4. State the properties of the active site. So the active site has a unique 3D shape, okay? And that is the answer. That's the phrase you need to use, the unique 3D shape. Okay, so make sure you're correcting and adding anything that you haven't got here. Question 5. State the product of starch digestion. So that's sugar or glucose. Question 6. State the products of protein digestion. Well, that would be amino acids. Question 7. State the products of lipid digestion. That's fatty acids and glycerol. You'll need both for the mark, please. Question 8. State the term used to describe an active site that has changed shape. So the phrase is denatured. So that's the term you need to use. Spelling isn't essential as long as it reads denatured. Question 9. Why can't the enzyme amylase break down proteins? Because amylase is specific to starch, okay? So you need to explain it's only going to work on starch, okay? Question 10. Suggest what would happen to the speed of an enzyme catalyzed reaction if the conditions were higher than body temperature. Explain your answer. So first off, it would slow down, okay? That's 1 mark. Anything to say it would slow down or stop. Secondly, the explanation is the enzyme would change shape or be denatured, okay, and that's the 2nd mark. So the 1st mark is it slows down or stops, and the 2nd mark is it's changing shape or it's denatured. Okay, so you get 2 marks for that one. So that makes this out of 11. Okay, so give yourself a mark out of 11 here at the bottom, please. If you've got 5 or less, then really you need to have another go at this and make sure you've learned some things here, okay? Because actually a lot of this lesson was learning, and you need to go away, learn some of those definitions, learn the table. And in fact, on top of this, I would recommend that you all look at the keywords page on page 31, where all the keywords are listed with their definitions. And again, this is part of your work that you need to make sure that you can explain what an enzyme is. Okay, what is amylase, what is lipase, what is soluble, insoluble protein, and what is the lock and key mechanism. So you need to make sure you can explain those. It doesn't have to be the exact words, but it does have to give the general idea. Okay, so that's the end of our lesson. And if you have any questions, please ask your teacher.